The gonads have two primary functions. The gonads function, number one, to produce the gametes, and number two, to produce the reproductive hormones. So those are the functions of the gonads. Now when we look at sex determination, we recall that in humans there are 46 chromosomes in a somatic cell, and in a haploid gamete cell there are 23 chromosomes. When we look at the diploid number, we have 23 pairs of chromosomes for 46. 22 of those pairs are called the autosomal chromosomes, and we have one pair of sex chromosomes. And of course, in males, there is an X and a Y, and in females, there is an X and an X. Chromosomal gender is determined by the fertilizing sperm, because the mother always donates an X, either this X, X1 or X2, but the father donates either an X or a Y, and so determines the sex of the individual. How does sexual determination occur under most circumstances? Well, if there is a Y chromosome in the zygote, as it develops, the Y chromosome has a gene, the SRY gene, that produces a testes determining factor protein, TDF, or testes determining factor protein. That testes determining factor causes the indifferent, undifferentiated embryonic gonadal tissue to go towards the testes, to go male. Once the testes are formed, they are going to be able to produce testosterone, and the testosterone will help continue the masculinization of the individual. The seminiferous tubules within the testes produce the sperm, spermatogenesis, as we'll see, and that's guided by some support cells called the Sertoli cells, as you remember. Outside of the seminiferous tubules, we have the interstitial cells of Leydig, and they produce the testosterone. The Leydig cells start producing testosterone about the 8th week of the embryonic period, and they reach the peak level at about 13 weeks. This masculinizes the embryonic structures. This is a critical time period for testosterone secretion. If testosterone secretion is too high or too low, or if the individual lacks receptors for testosterone, there can be differences that occur in this process. After the peak of testosterone during embryonic period at about 13 weeks, let's say, it slowly declines, and it eventually starts reasserting itself at puberty. The testes descend into the scrotal sac shortly before birth. Of course, in women and females, the ovaries stay in the pelvic cavity. Absence of the Y chromosome, the SRY gene, means an absence of the testes determining factor, and the indifferent, undifferentiated gonads develop into ovaries, the female gonads. So here is our X and Y, makes the fertilized egg, as you remember, is the zygote, and the Y chromosome with the testes determining factor, and eventually testosterone turns male. With the X, the indifferent gonads turn female. You recall that for females, there's two X's, but one X gets removed from every cell. So you have two variants of the X, but every cell you remove one of the X's. The remnant of the X chromosome that's removed can form what is called a bar body. A bar body kind of looks like a turkey drumstick, and they're often seen in some white blood cells like a neutrophil. This is a neutrophil that we see here. These are red blood cells, and these are platelets over here. So this is the nucleus, multinuclear nucleus. This little drumstick structure here is a bar body, which is the remnant of a inactivated X chromosome. So this would be blood from a female. In addition to forming the gonads, the reproductive structures also need to form. The male reproductive structures form from the Wolffian ducts, embryonic Wolffian ducts, and the female reproductive tract comes from the Mullerian ducts. The male would include things like the epididymis, the vas deferens, also called the ductus deferens, the seminal vesicles, the ejaculatory duct, and for female it would be the uterus and the fallopian tubes. You probably know that the fallopian tubes unfortunately have three different names, which are all used. They could be called the fallopian tubes, they can be called the uterine tubes, or they can be called the oviducts. It's all the same, but they have different names, so unfortunately, they all seem to still be used pretty frequently. This reminds us of the sexual determination. If it's XY, you have the SRY gene on the Y chromosome, and that then secretes testes determining factor, which causes the undifferentiated gonads to turn male, and you have the testes, the Leydig cells producing testosterone, which causes the Wolffian ducts to turn into the male reproductive system. Some of the testosterone is converted to dihydrotestosterone, which also stimulates the development of the penis, the scrotum, and the prostate gland. And the Sertoli cells within the testes not only support spermatogenesis later on in life, but they also secrete during the embryonic period Mullerian inhibitory substance, which causes the Mullerian ducts to regress and not form into the female reproductive system. In the female, we have the XX chromosomes, no SRY gene, and so the undifferentiated, indifferent gonads differentiate into the ovaries, and there is an absence of Mullerian inhibitory substance, so the Mullerian ducts are transformed into the female reproductive system, and the absence of testosterone, the Wolffian ducts regress. Now that's the normal course of action. There are disorders in this normal sexual development, and these disorders are called intersex disorders, and that can be classified into four major categories. The first two that we see here have the normal chromosome number, and the sex chromosomes would be either two X's or an X and a Y. These two categories, the disorders are usually due to either too much of a certain type of hormone at a critical junction, too little of a certain type of hormone at a critical junction, or an insensitivity to a reproductive hormone at a critical junction. The insensitivity is usually due to a lack or a decreased number of receptors for the hormone to bind to. So this causes the intersex 46XX and intersex 46XY issues that we'll look at in more detail in just a minute. In addition, we have what is called true gonadointersex, that used to be called true hermaphrodism. That is where you actually have a mixing of structures in one individual, of sexual gonads in one individual. And finally, we have complex or undetermined intersex. This is generally a differing number of chromosomes, a differing number of sex chromosomes. So we'll look at that as well. First of all, 46XX intersex. What's happening here? You have the normal chromosome number, you have the normal XX, but you have an excessive amount of androgen or testosterone. Just to clear it up, androgens are a broader category of male hormones that traditional male hormones, I guess you could say, of which testosterone is the most abundant. Estrogens are a family of hormones predominantly associated with women, where estradiol is the most prominent. We often use androgens and testosterone synonymously, although they're not exactly synonyms, and the same with estrogen and estradiol. Well, what's happening here is we get too many androgens or too much testosterone in a genetically chromosomal female. Most of the reason that we get this excess during these critical times during embryonic development is due to a disorder called congenital adrenal hyperplasia. You remember that in women, there is really only one source of androgens, and that is the adrenal glands, specifically the zona reticularis of the adrenal cortex. Women have androgen secretion throughout their life from the zona reticularis of the adrenal glands, perfectly normal. But during development, if there is an excessive amount of these androgens, testosterone, from the adrenal gland, it can cause some masculinization characteristics. And we'll see an image of this, but essentially it's due to a mutation coding for an enzyme is usually what's causing the problem. And so we'll look at that in a little bit more, flesh that out a little bit more in a minute. What about 46XY? So this is the chromosomes of a male, but the external genitals are incompletely formed, ambiguous, or clearly female. Why? What's happening here? Because in this case, the man's body is not responding to testosterone during this critical embryonic period. It's usually due to a lack of receptors for the testosterone to bind to. And so the testosterone is there, but it can't bind and to the target cells because the receptors are absent. Now this is congenital adrenal hyperplasia. What happens is, you remember, the hypothalamus produces corticotrophin-releasing hormone, which goes to the anterior pituitary, which produces adrenal corticotrophic hormone, which goes to the adrenal cortex and converts a pathway to convert cholesterol to cortisol and also cholesterol to androgens. The cortisol is produced in the zona fasciculata of the adrenal cortex, and the androgens are produced in the zona reticularis. In this abnormality, an enzyme, there's an enzyme called 21-hydroxylase. That's the most common one that's affected. You have on the gene a mutation, and so you don't make a correct enzyme for 21-hydroxylase. What that means is you don't produce as much cortisol in your metabolic pathway from cholesterol. So instead of getting proper amounts of cortisol, which would give you negative feedback to shut off ACTH, because you have decreased cortisol, you have less negative feedback, and so you keep secreting ACTH in excess, and that also causes the production of too many androgens. And when this happens during this critical juncture in the embryonic period, it can cause masculinization even though it's an XX female. On the other hand, you can have the XY male chromosome pattern, but lack the receptors for androgens or testosterone, in other words. And so an XY individual would have a phenotype that is female, the breast and the rest of the structures that are female. It still would respond to Müllerian inhibitory, and so it would lack a uterus and oviducts or fallopian tubes. The brain would also tend to be more feminized because, again, you're not having receptors for testosterone. Now, the rarest of these intersex disorders are true gonadal intersex. Here you have a mixture of both gonads from both sexes, ovarian and testicular tissue mix. You could either be XX or XY. The external genitalia appears ambiguous or may be female or male. This is very rare. Probably intersex is not that common, but of all the cases of intersex, this is maybe only 5% of all the cases of intersex. We don't know for sure what causes it, but these are the theories. One ovum that is ovulated divides, and then the two that are divided each are fertilized by a separate sperm, and then one would be male and one would be female, but then they fuse, the two zygotes, early in development. It can also have a mutation in the SRY gene, and finally it could be a chimera of two fraternal twins. A fraternal twins, you could have, in other words, a male and a female. They're no more alike than any other brother and sister, except they share the uterus at the same time. But early on, for reasons that are really unknown, they two fuse into one. So you have two fertilized eggs that are developing, one male and female, and then they fuse early on and form this mixture. Fortunately, this is rare because it's not it causes some major problems. Finally, we have the intersex disorders, which have an altered number of chromosomes. And we can have a number of these alterations. These are examples of some of these altered chromosomes. Turner syndrome happens in about 1 out of every 10,000 live births. This is where you're lacking one chromosome. There's one X chromosome, but there's no Y and there's no second X. So it's called XO. This results in a smaller stature, only rudimentary gonad development, underdeveloped external genitalia. Often, this person is sterile and have babies. Sometimes there can be a fairly mild mental deficiency associated with this as well. Klinefelter syndrome is there's two X's and a Y. This happens about 1 in every 2,500 live births. There's male genitalia, but may have female secondary sexual characteristics, such as more breast or mammary tissue. This is an individual with Klinefelter syndrome. Note that they have some mammary tissue, the more of a triangle pubic hair, which is more female-like, a small penis, sparser body hair. This would be Klinefelter. We can also have an X and two Ys, about 1 in every 1,000 live births. These individuals are taller, more prone to acne. They may be sterile, but not always. For years, it was thought that these individuals were more aggressive than the average person and more prone to criminal behavior. But they've done more good recent statistical studies and that found that that was not the case. There is not a greater number of people with XXY in prison, for instance. You can also have 3 X's, triple X, 47 chromosomes total, 1 in 1,000 female births. Most of the time, these symptoms are so mild that this is never even diagnosed. It causes very little issues. The main symptom is they're taller than average. So those are some of the intersex issues that can happen. Again, it could be due to a different number of chromosomes, or it can be due to a difference in the amount of hormone at a particularly critical time, too much or too little, or a lack of receptors for hormones. How do we regulate reproduction? In both male and female, we have the gonadotropin releasing hormone and from the hypothalamus, which can stimulate the two gonadotropins from the anterior pituitary, LH and FSH. Both of these hormones are really named for what they do in females. The luteinizing hormone for ovulation, for instance, and follicle-stimulating hormone stimulating follicular development in ovaries. But both of these hormones from the anterior pituitary have their normal function in men. It's just they're named mostly for what they do in women. LH and FSH are going to stimulate meiosis, spermatogenesis, and oogenesis. They're going to stimulate the reproductive hormones and maintain the gonads. This shows the hypothalamus with gonadotropin releasing hormone going to the anterior pituitary with FSH and LH, acting on the gonads, and then some negative feedback that occurs. This is not something for you to memorize. This is just to remind you that the reproductive hormones, by and large, are steroids. They're derived from cholesterol. They're non-polar. And here we see that there's metabolic pathways from cholesterol, and they can produce progesterone, androstenedione, testosterone, estradiol is the main type of estrogen. Chemically, testosterone and estradiol are not very different, but there is certainly enough difference for them to be distinguished. FSH and LH are high in the newborn, but then they fall to low levels until puberty. As the brain matures, including the hypothalamus, the gonadotropin releasing hormone from the hypothalamus is increased, and LH and FSH is increased. And this helps with the onset of puberty. This graphic is really illustrating that on average, females go into puberty at an earlier age than males. This is showing a change in growth, the height gain, between females and males, and essentially it's showing that females get their growth spurt earlier than males get theirs. The onset of puberty is correlated at least, to the amount of body fat and physical activity in females. We've noted that girls go into puberty earlier than they used to, on average. There's a number of theories to try to account for this. One is the theory that the percent body fat has increased, especially in developed countries, and this could lead to earlier onset of puberty because a hormone called leptin, secreted from adipocytes, fat cells, helps initiate the onset of puberty. You can see the name here, leptin. There's been other theories that suggest environmental contaminants, if you will, microplastics, and others might have estrogenic effects or other hormonal effects, and that might cause it. And so, what are we putting into our bodies, essentially, might be affecting this process. There's a number of studies looking at this, but we can't really definitively say what's leading to this earlier onset. But those are two of the main theories. When we look at the male reproductive system, the testes or testicles are the gonads, and they descend down into scrotal sacs outside of the body proper, outside of the pelvic cavity. And you know why. Why do we have to have the testes outside of the body proper?
So the sperm can live because they don't like high temperature.
They don't like high temperature. They don't like body temperature of 37 degrees or anything. They need a cooler temperature, maybe a degree or a degree and a half Celsius cooler. And so spermatogenesis does not occur nearly as efficiently at higher temperatures. This is one of the reasons that if a couple is trying to get pregnant and the man man is sitting in the hot tub all the time, that may not be the greatest thing, right? Because heating up the testes too much. Or they even, you know, if the underwear is too tight, maybe it's like, oh, you're heating up your testicles too much. So there's an issue there. Within the testes, there are a number of tubules called seminiferous tubules, and here we see them in the diagram here. Within the lumen of these seminiferous tubules, this is where spermatogenesis takes place. We have Sertoli cells that support this process of spermatogenesis. Outside of the seminiferous tubules, we have some Leydig cells, interstitial cells of Leydig, and those produce the androgens, testosterone predominantly. If we look at this histology slide, we can see the seminiferous tubules in cross-section undergoing spermatogenesis, and the interstitial cells of Leydig outside of the seminiferous tubules producing testosterone and secreting it into the blood. The sperm or spermatozoa that's a result of spermatogenesis goes from the seminiferous tubules to these rete testes here, and then they converge there, and then they go to the efferent ductules, and then they go to the epididymis where they're stored and get more mature for a while, and then they go on through the reproductive system, essentially through the vas deferens and on through there. We'll look at that a little later. This again shows us the spermatogenesis that's occurring in the seminiferous tubules with the support of the Sertoli cells. And again, they're going from the seminiferous tubules to the rete testes to the efferent ductules to the epididymis, and then they'll move on from there. All of this is outside of the body proper. Again, the hypothalamus secretes gonadotropin-releasing hormone, which goes to the anterior pituitary and secretes FSH and LH. LH in males stimulates the Leydig cells to release testosterone. FSH in males acts on the Sertoli cells to stimulate their aid in spermatogenesis in the seminiferous tubules. The Sertoli cells also produce inhibin, which inhibits FSH, negative feedback. Testosterone is also needed along with FSH to stimulate spermatogenesis. There's also negative feedback from testosterone to LH and to gonadotropin-releasing hormone. Testosterone initiates changes of the body at puberty, such as growth of the muscles, growth of the larynx, causing deepening of the voice, bone growth, hemoglobin synthesis, and of course, aiding in spermatogenesis. This is a little bit more inclusive list of the effects of testosterone in males. I've tried to highlight in these red boxes the major function of testosterone: spermatogenesis, negative feedback on gonadotropin-releasing hormone, negative feedback on luteinizing hormone, differentiation of the male accessory reproductive system, increasing male secondary characteristics. This is things like growing the beard, hair under the arm, the axillary, things of that nature. Protein anabolism, making more protein, more muscle, more bone growth. Sex drive, and it also stimulates erythropoietin, which stimulates also red blood cell production and hemoglobin production. So those are some of the major things there. How does the brain get masculinized? Well, it's a complicated process, and it's a bit counterintuitive. Although testosterone has some direct masculine masculinization effects on the brain. Most of the masculinization effects on the brain are due to estradiol. That seems opposite, doesn't it? Estradiol masculinizes the brain. Well, how does that work? Estradiol can cross over to the blood-brain barrier during the fetal period. Within the brain, there are neurons that convert testosterone to estradiol, aromatase. So in the brain you have estradiol. That estradiol, and we'll see it on the next slide, can then convert the brain toward masculinization. Well, then how come female brains aren't masculinized? Because female brains have alpha-fetoprotein in their blood made by the liver, and it binds to the estradiol and prevents it from binding to receptors in the brain. Males also have some lower concentrations of alpha-fetoprotein, so you don't have excessive estrogen effects from the mother during the fetal state. So essentially, females are protected during the embryonic period because they have a higher concentration of alpha-fetoprotein, which binds to the estradiol and prevents it from binding to receptors in the brain. Males have some alpha-fetoprotein, but a lower concentration, and in their brains they have estradiol can bind to receptors and cause it to masculinize. So here's testosterone with aromatase converting it to estradiol. It then binds to receptors and causes the formation of COX-2, which converts arachidonic acid to prostaglandin E2, and that masculinizes us. This is done in rat brains, but we believe it's a similar mechanism that's in humans. You just can't do these experiments in humans, without people protesting. But you could do it in a rat, and rats don't seem to mind, or maybe mind, can't do anything about it. So this has caused the masculinization. Some tissues, such as the prostate, testosterone is converted to dihydrotestosterone, another androgen. And the enzyme used to do this is 5-alpha reductase. If you have an excessive amount of dihydrotestosterone, you can try to inhibit the enzyme 5-alpha reductase to treat prostate gland cancer and male pattern baldness. So how are we feeling so far? I know a lot of this is, at least most of it, you probably reviewed, but you might maybe do at least bring it fresh to your mind, right? Some things may not be, may be new to you. Well, all right, let's review spermatogenesis. First of all, remember spermatogenesis takes place in the seminiferous tubules of the testes, and as you've mentioned, the temperature must be cooler than body temperature for spermatogenesis to work correctly and efficiently. The stem cells that will potentially form the sperm cells in the process are called spermatogonia. The spermatogonia can divide and multiply by mitosis, so you're just making more spermatogonia. Notice the 2N indicating they're diploid. As the spermatogonia go down the pathway to create sperm, the DNA replicates and a non-division differentiation occurs so that the spermatogonia differentiates into a primary spermatocyte. Now, a primary spermatocyte is diploid, but it has double the DNA. It's already there. So it has, in other words, 46 chromosomes, but they're double-stranded. Now, the primary spermatocytes undergo the first cell division to form two daughter cells called secondary spermatocytes. The secondary spermatocytes are haploid, as you see by the one end, but they're still double-stranded. So you're having double-stranded chromosomes 23 go into one daughter cell, and double-stranded chromosomes 23 going into the other daughter cell. So we count the chromosomes, there's 23, so they're haploid, but they're still double-stranded with the DNA. The secondary spermatocytes then divide again, each daughter cell forming two additional daughter cells for a total of 4 spermatids. Each of the spermatids is also haploid, and that's because your 23 chromosomes that are double-stranded now divide into single-stranded chromosomes. 23 going to this daughter cell, 23 going to that daughter cell. And so that's why you have haploid secondary spermatocytes followed by haploid spermatids. In the secondary spermatocytes, they're double-stranded chromosomes, haploid, and in the spermatids, they're single-stranded chromosomes. We all feel comfy with that? Look at that from General Bio or somewhere. The spermatids, without dividing, undergo a process called spermiogenesis, and that causes them to differentiate into sperm cells. This includes the growth of a flagellum or tail. For spermatogenesis, you recall that one spermatogonium or primary spermatocyte yields 4 mature spermatozoa. We'll see that in oogenesis, one oogonium produces only 1 mature egg, so there's some differences. This is just to remind you of the process here.
Spermatogonia are the cells from which sperm cells arise. The spermatogonia divide by mitosis. One daughter cell remains a spermatogonium, and the other becomes a primary spermatocyte. The primary spermatocyte divides by meiosis to form secondary spermatocytes. Secondary spermatocytes divide again to form spermatids. The spermatids differentiate into sperm cells.
The sperm cells are much smaller than the egg cells. This is an electron micrograph that's been colorized by a computer, showing an egg cell and a sperm cell, and showing some difference in the size. You remember that the sperm has a head, and over the head is an acrosome that contains hydrolytic enzymes that can help digest its way through the outer layers surrounding the egg and into the egg. And then it has a midpiece with some typical organelles, and then it has a flagellum or a tail for motility. There's the acrosome. Sertoli cells help the process of spermatogenesis. One of the things they do is provide tight junctions so that the area of the testes are protected. And then they aid in this development of the spermatogonia to the primary spermatocyte, secondary spermatocyte, spermatids, finding the sperm. The Sertoli cells are stimulated to do this under the influence of FSH and testosterone. This gives us a more inclusive list of the functions of the Sertoli cells. A cell barrier protecting the spermatogenesis process, provide nourishment to the developing sperm, androgen binding protein to bind to testosterone or androgen. They release some localized agents, paracrine agents, to help with sperm proliferation. Inhibin, which inhibits FSH as a negative feedback. Local agents that stimulate the Leydig cells to release testosterone. They phagocytize the effective sperm, and they can produce Mullerian inhibitory substance, which inhibits the formation of the female reproductive tract. The reproductive tract, here we see the scrotal sac and the testes. Of course, there's two, and then we see the epididymis. That goes into the vas deferens. The vas deferens is often called the ductus deferens now, so you'll often see that term. The vas deferens comes around and joins the ejaculatory ducts from each side. The seminal vesicles secrete fluid into that area. Then we go through the prostate gland with the ejaculatory ducts joining the prostatic urethra, and then the urethra of the penis carrying it out the rest of the way. Most of the seminal fluid that's ejaculated comes from the seminal vesicles and the prostate gland. There's two seminal vesicles and one prostate gland. The majority comes from the seminal vesicle, and then the prostate gland, the next in line, and a little bit comes from the so-called bulbourethral glands shown here. The bulbourethral glands mostly lubricate the urethra, but a little is in the seminal fluid. For our last little item today, we'll continue this tomorrow. A vasectomy involves going behind the scrotal sac on each side, making an incision and cutting out part of the vas deferens and cauterizing, burning the ends. This doesn't really affect the volume of the ejaculate. Most of it is this seminal fluid, but it means you're shooting blanks because you don't have any sperm being ejaculated. And it has to be done, of course, on both sides. You might ask, well, what happens to all the sperm that doesn't get any further? They just get phagocytized, just like the excessive sperm in the female reproductive tract gets phagocytized and taken away. So that's why this is called a vasectomy, and can often be done in a doctor's office. It's minor surgery, if you will. Of course, once you have a vasectomy, it's very hard to reverse the process, and so you generally don't want to have a vasectomy if in the future you may want to have more kids. Okay, well, so tomorrow we'll keep going with this. We have tomorrow, Wednesday, and we have Friday. And then we have test 3, a week from tomorrow. So remember, tomorrow we also have due the book report
there.